evaluators, welcome to today's Grade 10 Life Sciences show. Today we have a special revision lesson on structure and support in plants. We have selected highlights from lessons shown early in the term to help you revise and prepare for your June exams. You can download the notes for today's show from learnextra.co.za/live. Now it's time to get on with today's lesson. Please post your comments and questions on our Facebook page on facebook.com/learnextra or on Twitter @learnextra so we can help you with your revision.
What I've done is I've put a picture. Next to the picture, I have associated the functions. We call that an annotated diagram. And for those of you, all right, who maybe sometimes battle to study, it's always a good way, all right, in which to study. Don't always worry about the reams and reams and reams and reams of writing. Okay, you need to have a picture in your mind. You need to draw it. You need to label it. Take a double folio, draw, doesn't matter, don't have to be an artist, just to get the basic structure. Okay, so let's start off with the root. The dicotyledonous root and the monocotyledonous root, all right, will have the same function. A root's function is twofold. Number one, all right, it anchors the plant in the soil. So it needs to be a stabilizing force. And then number two, it absorbs the water from the soil. So when you look at its structure, remember its function. Its function is to provide support, anchor into the soil, and to absorb water. Let's have a look at the external structure. This is a diagram you need to become quite familiar with, the external structure of the root. As we see later in the program, one of our questions, right, that you can be asked on this, one of the more simple questions that can be asked is just to label this if you need to know the different regions. Okay, let's start off with, let me choose the color, let's have pink today. Okay, if we look at our root, remember our root is going to go into the soil. The roots burrow down and it goes into the soil. Now, because it's constantly going into the soil and burrowing, all right, and you need to think of soil. Soil has sometimes got stones in, as the sand can be hard, right? So as it goes down, what can happen sometimes is that the little tips of the roots, right, can be damaged. So now we need to have a protection factor. Now I'm going to go through the different regions and I'm going to explain to you what the function of each one is. Okay, let's start off over here. At the bottom over here, all right, we have a calyptra. It's called a root cap. Now that root cap is very much the same as a motorcyclist, all right, or somebody who rides on a bicycle wears a helmet for. That is going to be the protective structure. So as the root goes down, all right, as it's growing, behind this little root region over here, can you see there, this little region here? All right, that is the meristematic region. Now remember, what is meristematic tissue? Meristematic tissue divides by mitosis. So these are the baby cells, all right? They're the baby ones. They're the ones that are going to grow and mature. So you want to protect it. So this little root cap over here is protecting the meristematic region, the new cells that can divide by mitosis. These are ultimately going to become the ones that are going to differentiate into the different functions. So the calyptra, or the root cap, as it goes down, if that part gets damaged, the plant will just replace it. So what is most important is to protect these little new cells over here. Okay. The next region. So first of all, the root cap. Then we have our meristematic region, which we said that's our baby, our nursery. New cells are being made, and they now need to grow. All right? So a cell over here, once it has been made, will now go into the next section. And what does the plant need to do? It needs to grow. So it elongates. So here we have a region. Can you see over here? A region of elongation. So we would have a small little cell starting over there, and it would get bigger and elongate. And obviously, if a cell elongates, then the plant will grow taller, or the root will be able to extend. Okay, so there we've got the growing part. The next region, all right, is here, the root hair region. The root hairs are minute little epidermal cells that have got a slight bit of a change, but we're going to look at that just now when we look at the internal structure. The one function of the root was to absorb water. That's the function of the root hairs. They are responsible for the absorption of water. So we're going to have an area growing down over here, and over here, all these little microscopic structures, all right, they are going to absorb water, which is essential. Then when we look at the structure, the internal structure, you're going to then see how we need to just twinge it a bit, the normal epidermal cells, to make that function work. Then the next region, you will see it's called a mature region. When you did plant tissues, meristematic tissue, all right, you were told that it was an embryonic tissue. Embryonic means, all right, it is the early stage, it's at the beginning. Embryonic tissue can differentiate. Differentiate means to change into permanent tissue. So what permanent tissue do we find, all right, in our root? What are we going to find in this region over here, my mature region? All my plant tissues. I'm going to find parenchyma. All right, I'm going to find xylem. I'm going to find phloem. Those tissues are my permanent tissues. So I start off with a tissue that has a dividing function. It grows, and as it reaches its next level over here, it now starts to perform particular functions. That's what differentiate means. It changes to a cell that has a particular structure, a particular function, and that is your parenchyma, that is your xylem, your phloem, your sclerenchyma, your parenchyma, all of the plant tissues that you looked at. Okay, so when we look, you need to know the different regions, excuse me, the root cap or the calyptra, the meristematic region where we divide by mitosis, our region of elongation where our cells elongate and get taller, our root hair region where we obviously, all right, root hairs are produced for the absorption of water. Then, as those mature, the mature region, the cells differentiate, and they turn into our permanent tissues. If you have a look over here, you will also see something called a lateral root. Lateral roots are roots outside of the main taproot, because here you've got your main, all right, and there are side roots, if you want to call that. Lateral means to the side. And the lateral roots are there to help with anchoring the plant. They're also going to have root hairs for the absorption of water, but remember what I said, the two functions of the root: one, absorb water, and number two, to anchor. All right, so that's the external structure of a dicotyledonous root. Now let's have a look at the internal structure. If we have a look at the internal structure, I want you just to have a look at this. It's a simplified diagram. We call it a line diagram. A line diagram doesn't show any kind of cell detail. The only thing that it shows is that the root shows different regions where we can find different tissues. And we're going to look at 3 different regions when we look at the root. If you have a look over here on the outside, we are going to find the epidermis, or the dermal tissue. All right, the epidermis is the covering. It's always found on the outside of the plant, right? And it's there to protect the inner layers. Then you're going to see a nice big large region over here. All right, sorry, and that region is going to be called, all right, your ground tissue, or we can use the word cortex. Cortex, all right, the cortex giving large to a large amount. Then inside, right in the middle, right in the center, we are going to find our vascular tissue. So we've got epidermal on the outside, ground tissue or the cortex on the inside, in the middle. It's like the packing, you know, in between the in between cells that have just the packaging, like bubble, you know, that lasts like bubble wrap paper. That when you buy something that you need to protect, all of you love to, I'm sure, squish those bubbles. That's pretty much what the ground tissue is going to do. Then in the middle, vascular tissue, vascular tissue, remember, is xylem and phloem. Okay, simplified diagram. Now let's take a diagram and look at it, what it would look like with the different cells. Okay. Now you see why it's so important to have your foundation of your plant tissues. Because if you have your foundation of your plant tissues, you will then be able to recognize the different tissues that are found inside the root. If we have a look here, you'll see on the outside is our epidermis. Very important, right? My root hair, okay? My root hair over here. Okay, my root hair for the absorption of water. Here, look at these cells. Thin, largish. There's intercellular air spaces. Thin cell walls. Packing, packing, packing. What is my packing tissue? My parenchyma. Remember the most abundant plant tissue, my parenchyma. Then we see this very predominant central area. Okay, you need to be able to look at a diagram and be able to know instantly that it is a root. All right, you might get two diagrams of a root and a stem, and they ask you which one's which. So you need to be able to say, okay, this is the root. Number one region, it's going to have a root hair, and one of the most important features that you're going to see is right in the middle here. You're going to see the vascular tissue, and most specifically, what sticks out. All right. Okay, there's an X in the middle. All right, there's an X in the middle. And I always say to my kids, X marks the spot, because X is xylem. That's where we want to go. We always see an X for xylem, in the dicotyledonous root. Okay, let's have a look at each of those layers, little piece by little piece, and then you will see what I mean. Okay, this diagram should be familiar to you. I'm not too sure if you're going to do it now with the root, or your teacher might have done it with you when you did epidermal tissue, but this is epidermal tissue that is specialized. All right, if you have a look over here, this is the root hair. The root hair is responsible for absorbing water. Now, if you have a look at it, I want to show you the diagram underneath here. This is obviously a photomicrograph of the real thing. You will see that all these cells around here are normal epidermal cells, and these cells are the ones that are specialized. What's their function again? The absorption of water. So if we were to look at them, all right, here, can you notice this would be obviously, let's change the color quickly so that it fits more in. This would be the most obvious part. Right? And what is that? That is the root hair. It's an extension. And whenever we have an extension, we increase the surface area. All right. So this whole area, look at all these arrows. All of this area is exposed to the water in the soil. All right? If it was just a cell that looked like this, if it was a normal epidermal cell, that's all the surface area that would be exposed. But if we have an extension, what do? We have this whole area, right? And all of a sudden, we increase the surface area, more water can come in. What else do we have? Look here, a nice big vacuole. All right? What is my vacuole going to hold? It's going to hold water. All right, so the water can come in from the soil and go directly into that large vacuole. And then from there, we can move it through the plant to the xylem. Okay, so the epidermal cell is specially made just for its main function, and that is the absorption of water. Okay, see, most importantly. Also, what you will notice about the root, okay, I'm going to go back again. One label that you are not going to find when you get to the root, you will not find it because you do not need it, is a cuticle, right? A cuticle prevents the plant from water loss. So in other words, it's like putting Vaseline around your roots. Now all of a sudden you've got this waxy cuticle, and what can the water not do? Can't come in. So it can't perform its function. So that would be a bit stupid. Okay? So no cuticle in the root, because we don't want, all right, the water to not move in or out. We want the water to actually move in. I said to you in the beginning, a nice little easy question that we're going to start with. Study the diagram of the external structure of a dicotyledonous root. Okay, you've studied it, you know it. Okay, so if we have a look over here, I think we need to add a few. Let's add a few. One, that will be 1, 2, 3, 4, okay, 5. I think we have one missing. There we go. That's actually, that would be 1. Let's make that 2, that 3, that 4, that 5, that 6, and make that 7. Okay, a gremlin disappeared with the numbers. Okay, let's have a look the kind of question we can ask this. Nice and easy one to start off with. Right, this is not interpretation, this is learning. And you haven't learnt it from your book, you've drawn it on a piece of paper at home, you have labeled it, and in different colors, you have put all the different functions, so you are prepared for it. Okay, identify the parts labeled 1 through to 7. Let's start. Okay, 1, right, is our calyptra, or our root cap. If you can just remember root cap, that is fine. All right, what was the root cap protecting? Yes, our little babies, our meristematic, okay, not cells. What was it? A meristematic region. Each of these, all right, showed particular regions, particular areas. What was number 3? Elongation, the region of elongation. Once they were born, all right, well, born, you know what I mean. Once they divided, then they need to grow. What do they give rise to next? Here we go, number 4. All these little specialized structures are my root hair region. All right, I'm just going to put an R for region there. Then, what happens afterwards? Their cells change and they differentiate and become mature. All right, the mature region. Okay, all right. These are actually root hairs, yes, not the region, but the specific structure. And number 7 is this one at the top here, and that is my lateral root. I know it because I've studied it. That's my study. It's not interpretive. It is the study work. Okay, let's answer number B. Name the parts, functions, the functions. All right, what do they do of parts numbered 1 and 6? I'm going to do it on the diagram. All right, I'm going to use a different color. See if orange works. All right, what is the function of my root cap? It is to protect the meristematic, sorry, the nice and long, meristematic region. So it's to protect it. What is number 6? All right, they are the root hairs. What is their function? Absorption of water. Okay, I need to write slower. Water and minerals. Okay, remember, with the water comes minerals. So there we go. Right, remember what the functions were? Anchorage and absorption. Now we go into number C. How does the tissue in region 4 differ with the tissue in region number 2? Let's go. What are we looking at? Aha. Number 2, and here's number 4. What do you think the differences is? Before you even start, all right, let's put number 2 is what? Number 2 was the meristematic region. All right? And what did I say? What kind of tissue is it? It's embryonic. All right? They are square. Meristematic cells are very simple. Square cells, all right, regular with a dominant nucleus, because basically their only function is to divide. Now, what was number 4? The mature region. Okay, and what is the mature region? Permanent tissue. And what is permanent tissue? That's when tissue, the word I want you to use is differentiation. All right, differentiation. And what does differentiation mean? It changes into specific cells. So it goes from a cell that his function is to divide, all right, to a cell whose main function is quite simply to do specific things.
Okay guys, I hope you're enjoying this revision session. If you are struggling and need help, remember to post on the page or send an email to helpdesk@learnextra.co.za. Now it's time for a break, so don't go away. We'll be right back. Welcome back guys. I hope you are getting into the idea of revision. Please let us know how you are doing. I'd love to chat to you on the page or on Twitter. Enough of the chat. Let's get back to revision now.
I have got as you can see in front of me, I've got a small little tree branch over here that one of our producers got for me this afternoon. And what we're going to do today is we're going to look at how dicotyledonous plants can increase in width. Right, we're going to call it secondary thickening or secondary growth. I'm just going to show you the bit of the features on the stem first, and then after that we're going to go to the board and do a bit of the theory behind it. Okay, if we have a look at our stem, what we're going to notice most of all, and I just want to get around over here, is that our stem has got characteristic rings. And that's what we're going to look at when we see if a plant grows. We can actually tell how old, I won't say a plant, but a tree, needs to be a dicotyledonous tree. We actually can tell how old it is by counting the rings. But it's usually those pictures that you see when they have counted, all right, it all depends on how you cut it, and again, if you polish it up, etc., then you're going to see the rings much clearly. But I want you to notice this is tree bark, all right? This is the tree, and if you look carefully on it, and next time you go past a tree, I want you to have a look. You're going to see all these little bumps on it, okay? They arise from the surface. If I can turn it around there, okay, you get all these little bumps, and those are going to be lenticels, all right? Because you need to remember, unlike our herbaceous stems, the ones that are quite green and quite flexible, you need to be able to still breathe, all right? This tree trunk still needs to get oxygen, and it still needs to get the nutrients into it. So those little lenticels are going to be the breathing pores, okay? You will see last week when we did the stem, here are our lateral branches. There we go, our leaves. It's now autumn as well. Our leaves are starting to die. And here, all of these little leaf scars on it. But the most important thing that I want you to have a look at, if we go to the center again, all right, is here you can see quite clearly that there's a dark region and there's a light region. And if you can look carefully right around the outside, okay, there, that is, if you can next time if you see a little stem that's all right fallen to the ground, next time instead of just kicking it or picking it up and throwing it away, have a look at it, and you'll see over there that's going to be the cork. All right, so most importantly are the circles that are going to be formed in the stem. And if we look at this stem and we look at the one over here, all right, this is going to give me muscles today. Right, you can see that this one is thicker than that one. All right, so what is ultimately going to happen? This one can also grow much thicker. We're now going to go to the board, and we're going to start with our theory behind this secondary thickening. Now I want you to look carefully here. I asked you to pay attention to the different rings when we were looking at the tree. I said to you, secondary thickening is going to involve growing wider. And we're going to see that growth by a marked ring pattern. Now, this is my stem of a dicot. This over here is the stem of my monocot. So although we didn't do the monocot last week, all right, and you don't necessarily need to do it, look at why. I've put the question here. So why can dicots, all right, widen and not monocots? And if you just look at the picture, remember I said to you, we are going to look at rings. We are going to look at rings. Okay, there I have a ring. If I get to my monocot, I'm looking, what do I see? Okay, I see a ring on the outside, maybe, but what do I need? I need this ring on the inside over here. Where's my ring on the inside? There isn't one. So for me to make a ring, I need to have established at least a little bit of a beginning pattern, and we don't have that yet. Now, the secret lies in our vascular bundles. They are our secret weapon. Have a look here, okay? The vascular bundles, their structure, all right, their structure is the force behind the secondary thickening. I want you to have a look at these two over here. You'll see I've even put crosses and a tick on it. Okay, first thing, in order to be able to divide, if we have a look at the dicot over here, and you'll see, there we go, you need cambium. If I look over here at my monocot, all right, there I see the phloem, there I see the xylem, and I look for it, and question mark, there's no cambium. So there's no cambium present in my vascular tissue. So secondary thickening is making it wider, it's growing more, all right, especially in width, and I don't have the necessary tissue that is there, all right, to be able to do it. That's the first point. Second point, we talk about a open and closed vascular bundle. If you have a look at my monocot, have you look at this pink all around here. The vascular tissue is enclosed. It's like me, somebody putting cement all over me, all right? I can't move, I can't do anything. So that's exactly what's happened here. So in my vascular bundle of my stem, that sclerenchyma cap is actually stopping it from growing wider. But if we have a look here, all right, in my dicot, what do I see? I only see there's a cap at the top. That's it. A cap to protect. The rest of it, look around here, nothing. No sclerenchyma whatsoever. So, yes, I have a protective part on it at the top. So, on the outside, yes, it's protected. But because it's got no restricting tissue, it can divide as much as it wants to. And the last point, all right, what shape are they in? All these vascular bundles are in a circle, all right? When I come to my monocot, here's my root, all right? They can be anywhere. The vascular bundles have no pattern, but in my dicot, I have a definite round circle. So the structure of the dicotyledonous plant is essential to its function of increasing in width. So if we go over it again, there you'll see, okay, the vascular bundles must have cambium. That's why I put it nice and big. All right, they must be open, no sclerenchyma confining in them. And the last one, very important, okay, they must be in a circle. So just looking at the shape and the structure of it, we can see exactly what we are going to do. All right, so let's start. Okay. First thing, what is secondary thickening? It's when the plant has the ability to increase in width. Okay? Remember, primary growth, increase in height, going up and going down. Secondary thickening, going wider. Right, so again, remember, you must remember your terminology. Now, if you have a look again at dicotyledonous plants, especially, and we're looking at trees. Trees are big, all right? They can be there for hundreds and hundreds of years. So they have the time in which they are going to grow wider. If you look at a monocot plant, let's have a look at our millie, all right? The common monocot that we can use. If we look at our millie is an annual plant, all right? What does it do? It grows one season And then what does the farmer do? Whoever wants it comes and takes it out again. So it actually doesn't last long enough for it to actually need, all right, to undergo secondary thickening. And if you think of our trees, trees are heavy, they are big. The xylem and the phloem, especially the xylem, remember the xylem was hollow. The hollowness of the xylem and the hardness of it is going to make sure that we have a good supporting tissue. Okay, all right. So if we look here, I want to show you something here. Okay, this is the next important term that I need you to understand. I want you to have a look at this diagram over here. It's very simple to the ones that, all right, we showed just now. I'm going to draw a vascular bundle over here. So I'm not going to worry too much, all right, about the, I'm not going to worry too much about the cap. I'm going to want to concentrate on the phloem and the xylem, all right, and the cambium. Now, here comes the terminology again, all right? The cambium that is in between the two over here, all right, is called the intervascular cambium, all right? So what we're going to find over here, we have the cambium, all right, which is called fascicular cambium, all right? It means it's cambium within the vascular bundle, vascular bundle. Now, if you notice, each of these ones, let me go here, much easier, each of these ones are separate. Although they are arranged in a bit of a ring, right, we don't see a continuous ring. Now, what happens is, in between, here I'm going to use a different color pen, all right, in between these areas over here, there are little parenchyma cells. Okay, because remember, parenchyma, my packing. And what happens is those parenchyma cells actually change, right? They now become meristematic. So now they join up with their friends, their buddies, in the vascular bundle. So if I were to join it, okay, here I had my fascicular, here I had vesicular. Now I'm joining. So what have I got? If I have to join all of those, I now have my ring. Have a look at this diagram. All right, so it's showing you. There the vascular bundles were by themselves, and now, once that parenchyma becomes meristematic, all right, we call it, there's the word, interfascicular. Means in between the bundles, all right? That is your interfascicular cambium. So you had cambium that was by itself, and now you've formed it, so you have formed a whole ring. And that is very important.
Now it's time for a break, so don't go away. We'll be right back. Welcome back from the break. I know you can't wait for the term to end and the July holidays to start. And of course, that means winter school. This year, we will be repeating all the Term 2 Learn Extra live shows for you from the 24th till the 28th of June, from 9 a.m. in the morning till 7 p.m. at night. So don't miss out. Go to our website, www.mindset.co.za/learn/extra/winter school to get all the details and to download the notes for each lesson. But for now, let's get back to today's revision lesson. Over to you.
Now we're going to start to answer some questions on secondary thickening, all right? And I've started off with an easy-ish one, and then I'm going to go on to one where there's not so much studying involved, but a lot more of the skills, the graphs. Okay, looking at the diagram, sorry, let's see. First of all, you should notice that there's a ring, okay? So the first thing I look at, I see it's a dicotyledonous stem. Why? My vascular bundles are open, but most importantly, I can see that my vascular bundles, sorry, are arranged in a ring. Now, what is also what I've noticed, okay, is that the cambium has now joined. So my first thing, before I even look at this diagram over here, right, what am I looking at? I know that because that ring is present, my stem is now ready to undergo secondary thickening, which answers question number A. Which process, and that's the process, remember I said it's not a theory, all right, the process, and that is going to be secondary thickening, all right, or you can say secondary growth. They're the same thing. I'm just so used to saying secondary thickening. Now, the next one, simple study. Label 1 through to 8. Okay, so which is number 1 is my interfascicular cambium. All right? Because where's it pointing to? Let me just quickly rub out for you quickly. All right, if I just take that little area away so that you can see. Okay, what have we got over there? It's not the vascular bundle, it's the interfascicular. All right, now let's go over here to number 2. What do you think number 2 would be? Number 2 is the outside. What is that? Epidermis. What's underneath my epidermis? All right, number 3. My cortex, isn't that all my parenchyma? So this is my outer layer, all right, that is my epidermis. Then my next layer over here, inside the black is the packing, that's the cortex, all right. Where does number 4 go? Number 4 is pointing to the middle one. That is going to be my cambium. Sorry. Let me just make an arrow there and let me just rub out quickly for you. All right, my writing's much too big for the space I've allowed myself. Okay, so that's going to be my cambium. All right, number 4. Now number 5. It's a bump. It's in the middle. It stayed the same. You cannot now use, now be very careful, it's important what word you use here. You cannot use xylem. You need to be specific. It is going to be my primary xylem. And if I look at number 6, all right, it's the original, the very first. It is going to be my primary phloem. Okay, which now, because now if you notice when you go to number 8 and number 7, if you've labeled xylem and phloem, now all of a sudden you're going to label xylem and phloem again. All right, but they are two different labels. So the outside one, which one is on the outside always? Phloem. But it can't just be phloem. It was the one that came second. So it is my secondary phloem. Always on the inside is my xylem. But it wasn't the first. My little bumpies were first. All right. So what is it? It is my secondary xylem. Okay. So when you label the structure of a stem that's undergoing secondary thickening, please, it's on the outside, all right, primary phloem, secondary phloem. On the inside, primary xylem and secondary xylem. Okay, now the next question. Describe by using the process mentioned in question B, all right, okay, by using the label, numbers, and the names. All right, should actually have been, let's put that, the A. There we go, secondary thickening. Now they ask you to describe. When you describe, you actually have to write a sentence, a full sentence, all right? So what did we say is happening, all right, in secondary thickening? What happens? You need to start off with, okay, what happens to those parenchyma cells? They change, all right? They become meristematic. What do? They join with the fascicular cambium, all right? And what does it become? Interfascicular cambium. Okay, see, Now, I'm talking in Greek, yes, maybe I am, but I'm speaking biologically. I'm using words like parenchyma, meristematic, all right? The fascicular cambium becomes the interfascicular cambium. All right, now it's in a circle, so what does the interfascicular cambium do? All right, it divides and it lays down new xylem on the inside. And what do we call that xylem? Secondary xylem. All right, let me see if I touch here. No, I'm not going to get any more space. All right, and then what happens? We lays down new phloem. All right, and where's that new phloem? On the outside. Okay, and what do we call that new phloem? We call it secondary phloem. And what has happened to the plant? Very simple, it has grown wider. All right, so if you have a look at all of that, there's just terminology sprouting out from that answer. All right, it's, as I said to you, interfascicular cambium, secondary phloem, secondary xylem. All right, meristematic tissue. So when you talk about it, you do need to be somewhat specific. Okay, now we're going to start off with looking at the types of questions you can expect on the leaf. I know exams and tests are the least of your favorite, but at least we need to get you prepared. When we look at the question, all right, what the first do is when I look at it, you always need to use pictures. All right, here is a diagram. So if you know your diagram, and it's very similar to the one that we went through just now, already you need to start, all those terminology needs to go through your head. You need to think upper epidermis, lower epidermis, palisade mesophyll, spongy mesophyll, cuticle, xylem, phloem, intercellular S bases. All of that should be going through your mind. So let's take it one step at a time and see how we can answer these questions. Okay, study the following diagram. All right, of the cross-section through a dicotyledonous leaf and answer the questions that follow. All right, let's start off nice and easy. And we start off with a nice and easy question. All right, it's always nice to start off with the easy question. It gives you a little bit of confidence for you to carry on. Give the main function of this plant organ. All right, now if we have a look over here, if you read it properly, okay, it says leaf. You know exactly what you're looking for, right? It says there. It doesn't even tell you, study the diagram of a plant organ underneath, which sometimes it can. It's already telling you that it's a leaf. So when we look at it, we need to think. Now, there's a little bit of a catch here because you can't mention just any function. You need to mention the main function. And the main function, all right, you can remember, was photosynthesis. All right, 1 mark, nice and easy. Next one, point number B. These are nice, simple questions. These are studying questions. These are nice ones. If you know it, hey, you can score and get the marks. Provide labels for part number. Now you've learnt it. I know you have, all right? You've learnt it, and it's nice and easy. We go and we look. Okay, we want 1, 2, 9, 10, 12. So I look at number 1, all right? And that is, now be very careful here. Don't tell me epidermis. I want to know that it is the upper epidermis. All right? Or you could have some, as I said, some teachers say the adaxial or the abaxial. That would then be the adaxial surface. Okay? I think it asks for question number 2. Number 2, it's on top of it, and that is going to be my cuticle. All right, then we got number 9, 10, and 12. Number 9, I'm looking at it. What am I seeing? All right, here it is. This is a spongy mesophyll cell. So if it's a parenchyma cell, as we said, spongy mesophylls is a special type of parenchyma. That dark staining would be the nucleus. Because remember, tissues is made up of different cells. Then I needed number 10. You can have 2 here. You can use intercellular airspace, All right, but that's more for these ones over here. The one closest to the stomata over there, because it's pointing to that little space over there, that is going to be your air chamber. All right, your air chamber. So number 10 is going to be your air chamber. Number 12, look here, they've asked you for that whole structure. It's a bracket. So if it's a bracket, it's not asking for the whole, which would be the stoma. It's not asking you for the two side ones, which are the guard cells. It's asking you for the whole structure. And remember what we said? Guard cell plus stoma is stomata. There we go, my stomata. So just by knowing that it's a leaf and being able to label, if you are not studying using diagrams, you are going to battle. Take your notes, cover the labels, learn them. Certain things you need to learn off by heart. That is going to be one of them. Right? Sometimes looking at the cell, if you know your plant tissues, you can get away with it, but it's important if you know the labels. Okay, let's go into question number C. State the number. So now please be very careful. They've asked you to state the number. They haven't asked you, all right, to give the name of the plant or the tissue. Be very particular. If they ask for number, you only give the number. Okay, so that's the first plant. State the number of the region of this plant organ where you find diffusion of gases. All right, so where are we going to find the movement of gases? So if we go upwards, all right, if we take it up, where did I say? I'm going to quickly rub it out over here for you so that we can see it much clearer. Okay, where are we going to see the diffusion of gases? Which number? Okay, where? So here we've got a few options. We've got the pen again. I'm going to use pink just so you can see. We've got a few options. 10, 11, or 12. Okay, where does it come in and where does it come out? Your answer is number 12. Right, number 12, because that is your structure. All right, so the number is 12, okay, where you would find, okay, state the number of the region of this plant organ where you would find the diffusion of gases. There we go. I would also, I must be honest with you, I would also sometimes, if I was teaching and I saw that, I would give you the benefit of the doubt for number 10, because there you're going to have a diffusion gradient. But number 12, the stomata, would be your most accurate diagram. Okay, then again, number D, in which numbered part? Doesn't ask you to name it. There's a difference. Which number part does photosynthesis take place? Now we know by looking at it, which is the main tissue for photosynthesis. All right? Which number part does photosynthesis take place? Where do you think that it's going to take place? If you have a look here, you have two options. Which numbered part? That's the palisade mesophyll. But if we have a look over here, if we look at 7 and 8, they are the chloroplasts. All right? We now got a bit of a quandary, because 15 is the region, all right, and 7 and 8 are the chloroplasts. In which numbered part does photosynthesis mainly take place? All right, and we're going to go with number 15. All right, number 15, because that's the part, the palisade. Okay, mainly, there we go. Another word, mainly. That's why we know it is not the spongy mesophyll. Even though photosynthesis does take place, not mainly. Okay, now, number E, if we have a look at it, how is the part mentioned in the question D structurally adapted to the process of photosynthesis? What did we say just now? What was number 15? The palisade tissue. So, what did we say? Okay, it's for, now I want you to have a look very carefully at this mark allocation, because some of you are going to put thin. What does thin mean? Yeah, I'm going to give you one mark. All right? It says, how is it structurally adapted? All right? So I want you to tell me, it is thin-walled. All right? And now for your second mark, give me the reason. It is thin-walled, so gases, okay, or water can move freely. Right, what else? It's elongated. There we go, elongated, 1 mark. What does that do? Increase surface area. Okay, for sunlight. Right, what else does it have? It's got many chloroplasts. We can say packed with chloroplasts. Okay, what are those chloroplasts able to do? All right, chloroplast is the organelle for photosynthesis. All right, another way. Okay, so now you can't say, one thing you mustn't say is it is just beneath the epidermis. That is not a structural adaptation. All right, structural adaptation here. Here's the cell. All right, what does it look like? It's elongated, it's thin. All right, and there's lots and lots and lots of chloroplasts. So now we've taken the diagram of the leaf, all right, and now we've put it into questions. Can you label it? Do you know the different functions? The next question that we're going to go to is a bit of a higher level. It's going to step it up a bit. Okay, I think we've got one more. Oh, we still got a few more. Sorry, mine. Okay. Question F refers to gases. Which gas moves in? All right, at number 12. So if I just go up a bit, there we go. Which gas moves in? Now you need to remember that the leaf is opposite to you. So we breathe in oxygen and out carbon dioxide. What goes in? Carbon dioxide. What comes out? Oxygen. All right, so what comes in at number 12 during the day? Carbon dioxide. What goes out during the day? All right, oxygen. It said gases. Please, you may not use water. It referred specifically to the gases. Okay, label parts 3 and 4 and state what is transported by each. The word there, transported. So we know it's going to be my vascular bundle. All right? So, and it even says to it, hello, we even nice and kind to you and we say vascular bundle. The biggest problem here is you get the two confused. The nice big open ones, that's always your xylem. All right? So number 3 is going to be xylem. For 1 mark, don't forget the rest of the question. What did it say? Xylem and what it transports. Okay, it transports, TPT is my little thing for transport, water and minerals. All right, number 4, at the bottom here, they're usually much, all right, they're not as thick as xylem. And remember, in between my phloem, remember you had your phloem, and you always had a little companion cell next to it because it doesn't like to be alone. All right, there's my phloem. Okay, and it transports organic matter. Organic matter is a nice word for food. If you forget about organic matter, I'm just going to put a little arrow here. All right, sometimes it's a bit difficult to remember. You can just use the word food. Okay, let's go on to our last question. The part number 12 is involved in gaseous exchange. Okay, did you notice that will actually help you with your questions above? Name 3 gases that are exchanged through the opening at night. So what 3 gases are going to be exchanged through the opening at night? All right, what are they going to be? Oxygen and carbon dioxide. All right, although the plant, now it says at night. Night. That was very important. Okay. Okay, sorry. Okay, opening at 12. There we go. So number 12, if we look at it, it's here. It's our stomata. And what do we know? Stomata usually close during the night. Okay, so what happens is slight little bit. So now oxygen and carbon dioxide. Remember the plant, although it's not photosynthesizing, all right, what is going to happen? It's going to, all right, still its respiration is going to occur, and it's still then going to, all right, the carbon dioxide is going to be released. Now, what is the third gas? All right, we talk about, it's actually water, but it's water vapor. Right? Water gets lost in the form of water vapor. Okay, I'm sure you've all, like you breathe out, you've breathed against a window before, and it's made a nice little sign there. Okay.
We've come to the end of today's show. Thanks for joining me and for chatting to me during the show. Remember to visit our website, learnextra.co.za/live to get the notes and to watch the videos from the term. If you are stuck on any questions, send an email to helpdesk@learnextra.co.za and tell us where you are stuck. All the best for your exams.